So welcome to the laboratory floor of HFSC. This is where most of the casework is done. HFSC is about 200 employees and about 30,000 requests a year. Most of that flows through this floor at some point. And where this all starts is quality. We've got to get the right answer out there at the right time, but it has to be the right answer. So quality is the core of everything that we try to do in what is HFSC. So this is our R&D and quality lab. So we have a research and development group that helps validate methods, evaluate new equipment. They do a lot of work with our laboratory information management system, our core computer system that runs everything. So R&D does a lot of different things for us. Quality also is an extremely important division for us because they manage all of our audits and all of our assessments by accrediting agencies, but they also manage our blind quality control program. So we try to have about 5% of all of what the laboratory produces be what we call blinds. They're materials that we manufacture to look like cases, but they're things that we know the answer, what it's supposed to be. It's supposed to be a positive or supposed to be a negative or supposed to be whatever it is. Those are then put into the system before any of the analysts would know whether it's a case or one of these blind materials that they're handling, so that basically analysts are tested all the time. We test the entire system all the time as to if it's performing the way we expect. But in here is where we manufacture all those things. Because it's a real challenge trying to make things that look like cases. There's a lot of complexity to that, and the analysts are very good at spotting these things. So this is where it all starts, crime scene. I don't often come out to crime scenes, remember toxicologist, but evidence comes from scenes. Scenes are usually, almost always, chaotic, messy, they're in the field, they're unpredictable. But we try to overlay something objective, we have procedures in place, the unit is accredited because crime scenes are unpredictable. Best we can do is try and get the evidence off the scene cleanly, objectively, as systematically as we can, because everything else in the process is dependent on the evidence that we collect here. Our crime scene unit is 28 crime scene investigators, and that is to cover the 640 plus square miles of Houston. We have about 300 homicides a year. That is not anywhere near enough to do scenes like this that we get all the time. All right, so this is our vehicle evidence building. This is not something that we usually have on most of our tours, but since we have a virtual tour, we can do it this time. This is a facility near the main lab where we process vehicles as crime scenes. So we get vehicles from all kinds of crimes. They're used as part of the crime. They're a crime scene themselves. So we bring them over here in order to dust them for prints, measure bullet holes, photograph, document, all of those kinds of things. So this is forensic biology, or what a lot of people would think of as DNA. Main things that we ultimately come up with here are those DNA profiles that go into the national database, how we help identify people. This part of the laboratory is what we call screening. This is where we receive evidence in, we portion it out, we identify are there biological stains or places where there may be DNA for us to test on that sample. So in here, this is where we get that evidence, and we take photographic documentation of things, but you can see by how everybody's dressed in here, we're very concerned about both protecting our people from the evidence, but protecting the evidence from us. As we're all learning, we spew out stuff all the time that includes DNA from us that we don't want to get on the evidence. So we do a lot of work in here to try and keep us off the evidence in this part of the process. Right, so this is the next step in the process after we've sampled through what we're going to identify in a piece of evidence. So we've taken a cutting from an item of clothing or a swab. That then comes over here to extract the DNA out of that. We also do what's called quantifying. We determine how much DNA is there, because that determines whether or not we're going to go on for sequencing, actually determining what that sequence of DNA is. And all that stuff happens in here. So this is kind of the heart of the DNA laboratory, where we do extraction and quantitation and the amplification steps of the DNA processing. So to amplify the amount of DNA that's there, we may have one copy of DNA. So we need to make millions of copies of it so that we can see and understand what the DNA is there. So we do that through the process called amplification, where we take DNA and we unzip it. What we call denature it. We take the two strands and pull them apart. Then we add what are called primers, which are pieces of DNA that are designed to fit into specific regions of DNA so that we can then make another copy of that DNA from that primer. Then we stick it back together. So basically we've taken one strand of DNA and made two copies of it. And we do that over and over and over. It's an exponential process. Ends up with millions of copies of the DNA that we started out with. So once we have those millions of copies, we come into what's called post-amp, which is where we get the sequence and the information that actually becomes a result. So this is what raw DNA data looks like. It's not somebody's picture. It's actually a bunch of peaks that then somebody has to sit down and interpret. They have to go through and understand what all these mean, and it could be mixtures. It could be much more complicated than just the DNA from one person. In fact, most of the time, it's a mixture. Once we get that figured out, that's then the result that ultimately goes into the national database, what we call CODIS, or the Combined DNA Index System. And that's where you make the relationships between cases or between people in a reference to help make an identity of DNA that's left at a crime scene with an individual. So the next lab that we're talking about is latent prints. You start to get the idea that a forensic laboratory is actually like 7 different little businesses all incorporated into one lab because we have very different disciplines. So this is latent print processing. Part of what we have to do is be able to see the latent prints that are on things. So one of the things we do is what's called cyanoacrylate fuming, superglue. So we have these chambers that are built to control the humidity, and we basically turn superglue into a gas in there, and it settles on those oils and fats that are in a fingerprint and help make a fingerprint very visible, very fine detail. But that's what these chambers are for. So now that we've got the fingerprint visualized, we take images of it, we make a digital image of that because all the rest of our workflow is digital. So all of our examiners, because of COVID, now all work from home because everything becomes digital from that point. So an examiner will look at a latent print on their screen and look to identify all of those components of the fingerprint that may help them make an identification of that fingerprint to an individual. So part of the comparison process that we have is to run these fingerprints in what's called APHIS, which is the fingerprint database. So we have those digital images, we run them in that database, but it's like all the other databases, it doesn't just come back with somebody's picture and the address where they're at. An examiner has to then go through the list that's returned from that database. It gives likely candidates, but it doesn't give exact identifications. So once we get that, then we move on to an examiner comparing those and seeing if they actually can determine an identification. So toxicology. Got to admit, toxicology is probably one of my favorites because I'm a toxicologist. Toxicology for a crime laboratory are samples that come from driving under the influence of drugs and alcohol and samples that come from drug-facilitated sexual assaults. And so we're analyzing for the drugs that may have been impairing or may have been used as part of the commission of a crime. We get about 6,000 toxicology samples a year. That's a lot of drunks on the road. But in here, they go through what we call accessioning, or accepting that evidence in, making sure the evidence is correct. And then we analyze everything for alcohol. We do that by what we call headspace. So we measure the amount of alcohol that is associated with that blood. And then we analyze for drugs, all the various drugs that could be impairing. So this is the seized drugs laboratory. This is where we get pills, powders, green leafies, socks with green goo in them. Try to identify, is there a controlled substance there? Because that forms the basis then of the charges for a drug charge. So in this lab, we go through identifying, doing some initial testing on things before we set up to do the testing on the more specific chemical tests that we do. So one of the main tools that we use in seized drugs is what's called gas chromatography mass spectrometry. That's what you hear all these instruments doing. That lets us separate out all of the possible chemicals that are in a pill, say, and then that goes into a part of the instrument where we bombard it with high energy electrons, and we can see the parts of the molecule. That's what helps us identify those compounds specifically at the molecular level. This is our multimedia unit. Basically here, we're processing videos, and we process to retrieve information mostly off of these things, mobile devices. But almost all crime scenes anymore seem to have some kind of digital footprint and digital information that we need to get. So this is firearms. Actually, this is our vault for firearms where we have our reference collection. Firearms, we look to examine weapons to see if a fired projectile or casing came from a specific weapon, and we look to relate weapons between crimes. So why do we have this reference collection? We have these for demonstration purposes, spare parts to repair weapons, and other reasons where we would need to have a wide variety of weapons. But the point here is to relate weapons between crimes. First step in this process is what we call NIBIN, which is the National Integrated Ballistics Identification Network. It is the database of weapons to relate a weapon that we receive between crimes. It doesn't relate a weapon to a person, but it relates a weapon between crimes. Well, thanks for your time watching this. Sorry we can't have people through the laboratory as much as we would like these days. Hopefully that gave you some flavor of what we've managed to put together here, and I hope you appreciate the fact that Houston should be very proud of what we've been able to accomplish. We certainly are. We're pleased with where we've gotten, but we still have more work to do, and we look forward to continuing to make this a laboratory that Houston can be proud of. So thanks again.